All right, guys, welcome to Unit 5, Discussion 2, Transcription, Translation, and Proteins. We'll also talk a little bit about membranes and transport too. So proteins are a biological molecule. They're made of amino acids, and we use them to create enzymes, hormones, and other things for the body. Their shape is very much influenced by their function and vice versa. And we make these by taking DNA, we transcribe DNA to RNA, and then we translate it to the protein. So if DNA is our blueprint, then our protein is our final product. So transcription is going from DNA to RNA. We're still working with those nucleic acids. So in DNA, A to T, C to G. However, in RNA, thymine gets replaced by uracil, so it becomes A to U, C to G. When we're going from DNA to RNA, we're still staying in that same language, but we're making slight changes to help prep for that protein creation. When we get to the actual translation portion, we're taking that RNA, those instructions, we're reading them, and we're using it as a blueprint to build proteins from amino acids. So the reason we call it translating is because in essence, we're going from one language to another. We're going from nucleic acids and patterns and turning that into amino acids being put together for those proteins. And we use codons to do this. So codons take 3-letter patterns of nucleic acids and translate that into an amino acid. So you can see here on our chart, we got AUG altogether equals MET. ACG is the THR. So those are shorthands for the different amino acids. I'm not going to expect you to have memorized those or anything. Just be able to read the chart in order to translate a given strand. So our analogy here is using languages. When we transcribe something, we're going from one version to another, like spoken to written, or when we're prepping for translation, we'll make it more grammatically correct. So we might say, hey, I'm Jackie goes to hello, my name is Jackie, in order to really make sure that everything is clear, right? Then we translate, we go to that different language. So with our language analogy, we would go from hello, my name is Jackie to konnichiwa, watashi no namae wa Jackie desu. So that's what we're doing when we're taking this DNA to RNA and then RNA to protein. We're going from those nucleic acids to the amino acids and the proteins that it creates. All right, so let's transition into membranes. Membranes are especially important to cells. There are a couple different types of membranes. There are permeable membranes where things can pass through freely. There are semi-permeable where some things can pass through based on polarity, size, and other factors. And then impermeable means nothing can pass through. A semi or selectively permeable membrane is what we have in our bodies, and these are plasma membranes that allow some solutes and molecules to pass through via specific channels. We're like this because if our membranes weren't choosy about what goes in and out, then your cell wouldn't be able to maintain homeostasis and you wouldn't be able to survive. So if our membrane does need to be selective, how do we get those things across then? So to get things across, we use different types of transport. The first type we're going to talk about is passive transport, which is going to include diffusion, facilitated transport, and osmosis. The second type is called active transport, and it's got primary and secondary, and then vesicular. So passive transport is free. It moves molecules across with the gradient, and things always want to go from higher concentration to lower concentration until it's even. So passive transport utilizes that rule to help move things across, and it costs no cellular energy. If it costs energy, it's not passive. So diffusion is the most basic of passive transport. This is, again, that substance moving from high concentration to low concentration until it's equal. So you can see we've got a lot of our solute out here, and then it slowly passes through the membrane until they're even. This one is facilitated transport. So this is a type of diffusion, and this is where materials move across the plasma membrane with help from transmembrane proteins from high to low. It's still moving with the gradient, and it's still not using energy, but it is utilizing these carrier proteins, most of which have mechanical functions where when something touches them, they twitch open and close, or other things like that. Next is osmosis, and this is very specifically the diffusion of water through a semi-permeable membrane according to the concentration gradient. The interesting thing with osmosis is that water will always move from higher to lower concentration of the solute, but that doesn't mean that the water is being split evenly on both sides. What you see in the picture here is where you've got that solute, and the solute cannot pass through the membrane. So the water evens out in order to even out the concentration by putting more inside the membrane where more solute is. So when we talk about solutes, we talk about tonicity. And tonicity refers to the balance of the concentrations. So a hypertonic solution means your solution contains more solute and less water in reference to the cell. So tonicity is a comparison, right? If something is hypertonic, then you have to say what it is and what it's hypertonic to. So a hypertonic solution is hypertonic to the cell that is in the solution. When it is hypertonic, what you see here, if the solution contains more solute and less water, then the water is going to move out of the cell. And this is a bad thing because what you see here happening in these cells is they're shriveling up, right? All the water is moving out because the solution contains so much solute that the water is trying to even it out. An isotonic means that there is an even concentration from the solution to the cell. If you notice, you'll see water is still moving here. It's not that it's stagnant, it's just that it's moving freely at an even rate. And then a hypotonic solution is a lower concentration of solute in the solution as compared to the cell. And we've got water going in, right, as it's trying to even things out because the solute can't come out of the cell. So the water has to go in order to balance out that concentration. And you notice this is causing the cells to burst. All right, so we talked about passive, which is free. Now let's talk about active. Active costs energy, and we pay that energy cost through the use of ATP and other carrier molecules. And this is going to move your molecules against the gradient. So this is going to take things from lower concentrations to higher concentrations. You might say, okay, but why would you want to do that? Well, sometimes you need to build up a high gradient of potassium or another mineral or material inside the body in order to trigger another cascade. So primary and secondary active transport, these use our sodium ion pumps and gradients to move things across the channel. We're not going to go into any more detail than that. Just know that it's the sodium ion pumps. The other form of active transport is vesicular transport, and this uses endo and exocytosis. I'm including this on here, but we're not going to go super into detail in this year. Endo is into, and it uses those vacuoles and those vesicles we talked about to essentially encapsulate materials outside the cell and draw them in after closing behind it. It's very important that you note that it's closing behind because the cell is doing this without exposing its insides, right? Obviously, the cell can't just poke a hole and let everything go flying in and out. So the way these work is by attaching to the membrane or pinching off the membrane. We can pull things in or let it out. All right, that's all for this one. Catch you guys next time.